Welcome back to Chapter 3, Genetics. We're picking up where we left off from Chapter 1. We talked about Mendel and his monohybrid crosses, and now we're going to continue with dihybrid crosses. So remember that monohybrid crosses involved individuals that varied only by one trait. And a dihybrid cross, we're looking at individuals that differ by two traits. So in this case, we've got yellow round peas versus crossed with individuals that have green wrinkled peas. So here we're looking at color and texture. Okay, two different traits. So the purpose of Mendel doing this particular experiment, these dihybrid crosses, is he was wondering, are these different traits inherited independently, or do they follow each other? So if he crossed these two plants, is he always going to get yellow round and green wrinkled offspring? Or is it possible that you could get yellow wrinkled seeds, round green seeds, or in combination? Now what he found when he crossed this yellow round individual with a green wrinkled individual in the parental generation, what he found was that the F1 plants contained yellow round seeds. And because he had already done the experiments with the monohybrid crosses, he determined that these were the dominant alleles. These were the dominant factors. But what he found in the F2 was that he saw all phenotypic combinations. So as I mentioned before, the yellow round, the green round, yellow wrinkled, green wrinkled. So what this means is that he concluded that each trait is controlled by a different gene, and that the two alleles, or the two genes, are inherited independently. If he had gotten, again, all yellow round and all green wrinkled, that would mean that they are inherited together, but they're inherited independently, and therefore he's able to see all four phenotypic combinations. Now, interestingly, he found these phenotypic combinations in a 9:3 to 3:1 ratio. So let's explore that a little further. So if we analyze these dihybrid crosses, looking at the gametes, so if we start with a yellow round seed and a green wrinkled seed, so here the color is denoted by a G, uppercase for dominant, lowercase for the recessive. I'm losing my cursor. There we go. Lowercase for the recessive. And so this particular individual can only pass on the dominant G or the dominant W to their offspring. So the gametes can only contain the G and the W. In this case, this individual can only make gametes with a lowercase g, the recessive G and the recessive W. And so when you have the F1 generation, these F1s are inheriting a dominant G and a lowercase g, a dominant W and a lowercase w, and so they're heterozygous for both traits. What's interesting is what comes next, and then it gets a little complicated, because now you have these hybrids here, and they can produce many different types of gametes. They can produce an uppercase G, dominant G with a dominant W, a dominant G with a recessive W, recessive G with a dominant W, recessive G, recessive W. These are the different combinations of gametes that they can perform. Form. Now, the idea here that Mendel realized was that the alleles are going to segregate during the formation of the gametes, and they're going to segregate independently from each other. And so what this means is that the gametes can inherit an uppercase G, and it can inherit that with an uppercase or a dominant W as well as a lowercase w. It doesn't matter. It's the same chance that that's going to happen. And this is what Mendel called his principle of independent assortment, and we'll get back to that in a little bit. Oh, actually I want to go back for a second. So again, we can make this square here, which is called a Punnett square, which we'll also get back to in a little bit, where we have the four possible gametes, similar to the square that we saw previously with the monohybrid crosses. And if we marry up these boxes here, we find the different genotypes for each possible combination of gametes, as well as the possible phenotypes. And this is where he sees his 9:3 to 3:1 ratio in the phenotypes. keeping in mind that these phenotypes here, because each of these individuals in this particular category, they are displaying the dominant phenotype for each trait. So keeping in mind that if you are heterozygous, you are going to display the dominant phenotype, and that's why these 4 different genotypes will give the same phenotype. All right, so let's look at his observed versus his expected dihybrid results. And what you're seeing here is what he expected to see. This 9:3 to 3:1 ratio is pretty much what he observed to see, or what he did observe. So this leads us to Mendel's principles, and we looked at one of them already in part 1. So his first principle is a principle of dominance, and again, that's in the part 1 video, not in this video. And this principle states that in a heterozygote, one allele may conceal the presence of another. So you have a dominant allele which can conceal the expression of the recessive allele. Mendel's second principle is his principle of segregation. This means that in a heterozygote, there are 2 different alleles, and they're going to segregate from each other during the formation of gametes, and they're going to segregate independently. The gametes are, any given gamete is just as likely to inherit one allele, or be formed of one allele, as the other. And his last principle is the principle of independent assortment. And in this, he concluded that the alleles of different genes segregate, or as we sometimes say, they assort independently of each other. And this is what he was able to illustrate in his dihybrid crosses, that these different genes are not going to necessarily follow each other, that you're just as likely to have green seeds that are wrinkled or green seeds that are round. Now, these are Mendel's 3 sort of famous principles or laws that refer to genetics and how traits are inherited from parent to offspring, or passed on from parent to offspring, and these still hold true today. So I find that pretty amazing. These are published in 1865, and we still follow them today. All right, so moving on to the application of his principles. So if you're following along here at the bottom, I have this little timeline here. We're now on to sort of the second part of the chapter, the application of Mendel's principles. So he did these experiments, he determined the results. Well, now let's see if we can predict what the outcome of his crosses are. Now, there are 3 different ways to do this. One is a Punnett square. We've seen that already with the monohybrid and dihybrid crosses. Then the second 2 are the forked line and the probability methods. And these are useful for when you have a dihybrid cross or greater. So what I mean by greater is a trihybrid cross, or maybe even more than 3 traits. Whereas a Punnett square, really only useful or practical with a monohybrid and dihybrid cross. So here's a Punnett square. We've seen this figure before. And essentially, again, really only useful for monohybrid and dihybrid crosses. And the problem is that once you get to more than 2 traits, the Punnett square just gets really big and really complicated, and it's really not practical. So let's move on to the forked line method. And here we're going to talk about something called an intercross. And an intercross is really just when you have 2 different organisms or 2 different individuals that differ when they're crossed with each other. So a forked line method is really just a branching line diagram, as you see below. And here we're seeing a trihybrid intercross. So we're looking at 3 different traits, and we're crossing 2 individuals, each of whom is heterozygous for each of these 3 traits. Now, if we think about these heterozygous individuals, if you were to cross, let's say, an individual heterozygous for this trait with an individual heterozygous for that trait, and only look at this trait, what you would expect is a 3-to-1 phenotypic ratio. And we know that from Mendel's monohybrid crosses. So what we can do with this forked line method is just simply treat each of these 3 as essentially 3 different monohybrid crosses, and look at that in order to determine the overall expected phenotypic ratios in the offspring. If we're going to use this for a test cross, which is essentially when you're crossing organisms that may be homozygous for a particular trait. Again, the same thing. You're going to analyze these as 3 monohybrid crosses. Alleles for the genes in these, if you take these individuals, there's a 1-to-1, there's essentially a 50% chance that the gamete will have a dominant allele or the recessive allele. That's what I mean by a 1-to-1 ratio. Whereas the homozygous parents can only pass on the recessive alleles. Therefore, the phenotypes in the offspring are really going to be dependent upon the allele inherited from the heterozygous parent. Because if the heterozygous parent passes on, I keep losing my cursor, sorry about that. If the heterozygous parent passes on the dominant trait, you're going to see the dominant allele, you're going to see the dominant phenotype. If the heterozygous parent passes on the recessive trait, you're going to see the recessive phenotype. Okay, the probability method. Now this is where it might get a little complicated. So we can think about probability. I like to start thinking about it in terms of flipping a coin. So if you're going to flip a coin, you can think about what's the probability of flipping either a head or a tail. All right, well, since there are only two possibilities, the possibility of getting either a head or a tail is going to be 1 out of 2, or let's say 50%. But what if you flip the coin 2 times? What's the probability of flipping either a head or a tail both times? So essentially, what's the probability of getting a head 2 times or a tail 2 times? And that's where we're going to think about these different probability rules. One is the multiplicative rule, or I like to call it the AND rule, and the other that we'll talk about in a little bit is the additive rule, which I like to refer to as the OR rule, but we'll come back to that. All right, so the rule is the multiplicative rule. If you have 2 events, in this case a head or a tail, or A or B, if they're independent of each other, which means when you flip a coin, it doesn't matter, you have equal chance of getting one or the other, the probability of them occurring together, which means that's where my AND comes in. That's where my AND rule. That's why I call it the AND rule. The probability of having those 2 events both occurring is going to be the probability of the first event, which, let's say tails, times the probability of the second event, maybe getting tails again. Okay, so the way this refers to genetics is what's the probability of the offspring inheriting, let's say, the dominant allele from a heterozygous parent? Let's think about that. And also, if you have a cross of 2 heterozygous parents, what is the probability of getting offspring that are homozygous dominant or homozygous recessive? So let's talk about each of these 2 things. So what's the probability of the offspring inheriting the A allele, or the dominant allele, from a heterozygous parent? Well, the probability of inheriting any given allele is 50%, because if you have a heterozygous parent, there's a 1 in 2, or a 1 out of 2, 50% chance that the gamete will contain the dominant allele or the recessive allele. If you have, however, a cross of 2 heterozygous parents, what's the probability of getting a homozygous dominant offspring? Well, keep in mind, in order to get a homozygous dominant offspring, the offspring must inherit the dominant allele from one parent and the dominant allele from the other parent. Again, here's our AND rule. So the probability of getting the dominant allele from one parent is 1/2, 1 out of 2. The probability of inheriting that allele from the other parent is also 1 out of 2. So the probability of inheriting them both, so the probability of both things occurring, is the AND rule. You're going to multiply. 1/2 times 1/2 is 1/4. And of course, the probability of getting a homozygous recessive offspring from the same parents is going to be, the probability is going to be the same. All right, the additive rule, the OR rule. So what if you have 2 independent events, A and B, the probability that at least one of them is going to occur, so perhaps OR is the sum of the 2 probabilities. There's a more complicated definition, but for our purposes for genetics, we're going to leave it simple. All right, so for example, if you have 2 heterozygous parents, what is the probability of having a heterozygous offspring? Well, the probability of getting the dominant allele from one parent and the recessive allele from another parent is 1/4. Okay, because dominant is 1/2, recessive another 1/2. You multiply those. But there's 2 ways to get the heterozygous offspring. You can get the dominant allele from one parent, from the mother, and the recessive from the father, or the recessive from the mother and the dominant from the father. So in this case, because there are 2 possible explanations, 2 possible scenarios, it's an OR situation, so we add them, we get 1/4 plus 1/4 equals 1/2. This concludes part 2. Please join me again in part 3.